Good morning, everybody, and good morning to yet another super cool and super awesome session of Class 12 Biology. And today is a big surprise day for us and also for you. So let's see what it is. So here is a surprise. It's Hearts, the exclusive channel, is being renamed as BioPoint, the stream, the world of bio. So here we are going with the launching ceremony of BioPoint Stream, the world of bio. Let's crack NEET 2021. Okay, I'm excited to take the very first lecture on this particular channel, BioPoint, Stream, the world of bio. Okay, so let's continue with our session for today. And as you all know that today's topic is a very important topic, that is molecular basis of inheritance, and we are moving with the lecture number 1. Okay, so here we have the BioPoint Stream, the world of bio, starting with the lecture number 1 on the very important chapter, molecular basis of inheritance, which constitute the chapter genetics, that is principles of inheritance and variation, molecular basis of inheritance, and the last chapter, evolution, just contributes to nearly 34 marks, 35 marks of your NEET paper. So very important chapter, direct but tricky questions comes from all these chapters. So without wasting much of our time, let's move with the very interesting chapter number 6, molecular basis of inheritance, from class 12 biology. Let's start. Yes, nucleic acids. Okay, nucleic acids, there are 2 kinds of nucleic acid. Okay, as you all know, the basis of this chapter, nucleic acid are of 2 kinds, that is DNA and RNA. Both are nucleic acid. So in the majority of the organism, DNA is the genetic material. In most of the organism that we know, DNA is the genetic material. Okay, even though the first genetic material that was discovered was what? It was not DNA. The very first genetic material that was discovered was RNA. But DNA occupies or plays a major role. Okay, this is the genetic material in most of the organisms. So DNA and RNA, right? DNA and RNA. The only difference between there are so many differences, and the only difference that you might know is that DNA may consist of a 2-deoxyribose sugar. Okay, 2-deoxyribose sugar. Whereas RNA consists of a ribose sugar, and there are much more differences awaiting for you from this chapter. So RNA is the genetic material. RNA is not something which can be ignored. RNA is the genetic material in some viruses, right? Coronavirus, they have the material, genetic material is RNA. Okay, you have to study about the process, different processes by which DNA converted into RNA, RNA converted back to DNA by the enzymes. All these things you have to study. Transcription, translation, everything is there in this chapter. Even though RNA is a genetic material in some viruses, it has some more functions. It functions as messengers. It functions as messengers. Moreover, it can function as an adapter molecule. It can function as an adapter molecule, as a messenger, structural. Okay, it is the it acts as an adapter, then it acts as a structural, and the catalytic molecule. It can also act as a catalytic molecule in some or the other case. So all these points from this particular slide is very important. Okay, once again, recap everything. Okay, nucleic acid, that is DNA or RNA, it is of 2 kinds. Nucleic acid is of 2 kinds, that is DNA and RNA. Okay, They are the building blocks of genetic material. Nucleic acid are the building blocks of genetic material, which has two components, DNA number 1 and RNA number 2. DNA is a genetic material in most of the organism, whereas RNA is the genetic material in some viruses, and RNA mainly function as messengers. It can also function as adapter, structural and catalytic molecule. Okay. So next one, structure of a polynucleotide chain. Structure of a polynucleotide chain. Polynucleotides are the polymers of nucleotide. What are the main components of a nucleotide? The nucleotide has a components that is a sugar, a nitrogen base, a pentose sugar, and a phosphate group. Okay, and a phosphate group. So sugar, pentose sugar, nitrogen base, and a phosphate group. These three components constitute a nucleotide, and several nucleotides together combine to have a polynucleotide. Several nucleotides combine together with the help of a particular type of bond. You may call that bond as phosphodiester bond. Phosphodiester linkage or phosphodiester bond. Okay, is it clear? So we have two types of polynucleotides, that is DNA and RNA are two types of polynucleotide. DNA and RNA are two types of polynucleotide. Suppose if you check at this particular diagram, if you take from right from here to here, you may call it to be what? A polynucleotide. A polynucleotide. And if you case the one case or one column, one box, you call it as nucleotide number 1, nucleotide number 2, nucleotide number 3, and nucleotide number 4. 4 are shown. So if the nucleotide number 1, it runs from 5' phosphate and it will ends in the 3', and the complementary will start from 3' to 5'. That is the structure with the DNA molecule, right? If this particular coil starts from the polarity 5' and ends in the polarity 3'. Another nucleotide sequence having the polarity 3', 5'. Okay, these are complementary to each other. These two are complementary to each other, and what you can observe is that this is the 3', 5' molecule or the DNA sequence, and it may combine with the another sequence. Okay, since they are complementary 5', 3'. This is a very, very important point. Okay, the two DNA sequences are complementary to each other. The two DNA sequences being complementary to each other. They just bind together to have a nucleotide or a DNA, double-stranded DNA. Okay, we'll discuss about it in the later of this class. Double-stranded DNA, and this model of double-stranded DNA was proposed by Watson and Crick. This you have studied in your class 11 biomolecule chapter cell. Okay, Watson and Crick put forward the double helical structure of DNA. This is a DNA having the structure 5', 3'. Another DNA will bind together to it to form a cross-linked structure having the complementary polarity. Okay, so DNA is a long polymer of nucleotides or deoxyribonucleotides. That is the difference between an RNA and a DNA, right? So in a DNA, you may observe what? 2 deoxyribose. In a DNA, you may observe 2-deoxyribose, and in case of an RNA, you observe a ribose sugar. Okay, a ribose sugar is only observed. Ribose sugar. Okay, so nucleotides and nucleoside. So before that, I'll explain one more thing. That is, this is the nitrogen base. Since you have studied it one time in your 10th standard or in your 11th standard biomolecule chapter, I'm teaching it like this. Okay, you know this. That's why I'm taking the important points in one particular slide and just telling everything. Okay, I'll just give you the mind maps. Okay, so this is a pentose sugar. This is the pentose sugar, and this is the nitrogen base. Nitrogen base and the pentose sugar. Nitrogen base and the pentose sugar, they are known as nitrogen base and the pentose sugar. They are known as what? Nucleoside. Nucleosides. They are known as what? Nucleoside. And when a phosphate group is added to this particular nucleoside, you will be calling it as to be what? Nucleotide. Is it clear? So, next one. Okay. Structure of a polynucleotide chain. Structure of a polynucleotide chain. Polynucleotide chain are the polymers of nucleotides, and you have two types of polynucleotides, DNA and RNA are the two types of polynucleotides. Okay, so a polynucleotide has three components. That is, first one is a nitrogenous base. The nitrogenous base are of two types, that is purines and pyrimidine. Okay, purines and pyrimidines. A pentose sugar, that is a ribose in RNA and deoxyribose in DNA. I'm not saying this is important, that is important, because everything in this chapter is important, right from the first point of your NCERT textbook to till the last point, that is DNA fingerprinting. Each and every point is important. Is it clear? And a phosphate group. Okay, anyway, I'm telling this is a previous year NEET MCQ from this particular chapter. Okay, this nitrogen base as well as this sugar together constitute nucleoside. What was the term? Nucleoside. The nucleoside plus phosphate group. Nucleoside plus phosphate group together forms a nucleotide. Okay. So next one. So nitrogen bases are of two kinds. Nitrogen bases are of two types. That is nitrogen bases. I think the color is not visible. Yes. Nitrogen bases are of two kinds, that is purines and pyrimidines. Purines and pyrimidine. Purines are again classified into two, that is adenine and guanine. Pyrimidines are classified into cytosine, thymine and uracil. Thymine and uracil, thymine is present only in DNA and not in RNA. Whereas uracil is present only in RNA and not in DNA. The structure is not so important. You won't be asked about this. Given the structure and ask you to name it, you won't be asked. Okay. What is the other name of thymine? It is 5-methyl-uracil. 5-methyl-uracil. Look, additional methyl group is present in case of thymine in addition with the uracil. That is 5-methyl-uracil is present in case of DNA and uracil is present in case of RNA. Previous year AIMS MCQ. AIMS, NEET, AIPMT. Every important question. Okay, you will be asked these questions. Purines and pyrimidines. Sometimes problem also comes from this chapter and we'll discuss about the problems. Okay. So structure of a polynucleotide chain. Okay, we are going to discuss about the structure of a polynucleotide chain. A nitrogenous base. We have already learned that nitrogenous base is linked to the pentose sugar. The nitrogenous base is linked to the pentose sugar with the help of N-glycosidic linkage. N-glycosidic linkage. Okay, nitrogen base plus pentose sugar together forms what? A nucleoside. Okay, a nucleoside. So this particular bond is what I'm highlighting, the bond with the blue color. So this particular bond that connects a pentose sugar with a nitrogen base is known as what? N-glycosidic linkage. Is it clear? So if you just look, you will see that these are the nitrogenous bases. This green line joins the nitrogenous bases, right? And this blue and the red color, these two represents what? The backbone. The backbone of DNA or nucleotide sequence. Backbone of a DNA or RNA is constituted by the pentose sugar and a phosphate group. Pentose sugar and a phosphate group. Pentose sugar, okay, so phosphate group and the pentose sugar constitutes the backbone. Is it clear? Okay, so let's move with the next one. So we have already told that the nucleosides are formed. Okay, this is a nucleoside. Okay, please don't think that I just left it unimportant. It can also be asked for your NEET exam. Okay, so this particular structure that is formed by the pentose sugar and the nitrogen base is known as nucleoside. Is known as nucleoside. This nucleoside, that is, there are 4 nitrogen bases such as adenine, guanine, cytosine, thymine, etc. Right? Uracil, etc. So all these can form nucleoside. For example, adenine, in case of RNA, it forms adenosine. In case of guanine, it forms guanosine. C, cytidine, not cytosine, okay? T, thymidine and not thymosin. U for uridine. In case of RNA, in case of RNA, you won't get this thymine. Thymine in case of only DNA. Okay, DNA. So for case of DNA, it forms 2-deoxy adenosine, 2-deoxy guanosine, 2-deoxy cytidine and 2-deoxy thymidine. It will form like that. Okay, since the sugar is 2-deoxy, exactly over here. Look, nucleosides in RNA and nucleosides in DNA. Nucleosides in RNA is adenosine, guanosine, cytidine and uridine. Deoxyadenosine, deoxyguanosine, deoxycytidine and deoxythymidine. Look how it is arranged. Adenine, guanine, cytosine and thymine or uracil. Uracil in case of RNA and thymine in case of DNA. Okay, so let's move with the next one. So this is the fundamental basic structure of a polynucleotide chain or a DNA sequence, or either can be nucleotide sequence. Okay, it consists of a phosphate, it consists of a pentose sugar, and it consists of a nitrogenous base. So nitrogenous base plus sugar, you call it as what? Nucleoside. This nucleoside plus phosphate together giving what? Nucleotide. Okay, nucleotide. So nucleotide from side plus phosphate. Nucleoside plus phosphate together giving nucleotide. Okay, nucleotide is formed. So nucleotide is a fundamental basic unit of a DNA sequence, and several nucleotide sequences continue together to have what? A polynucleotide sequence with the help of phosphodiester linkages. Okay, so in RNA, each molecule has an additional OH group at the second position of ribose sugar. Okay, what is the main difference or what makes RNA and DNA differ? The one point is this. Okay, one point is this. RNA, in RNA there is an additional OH group at the 2' end of the atom. Not atom, in the sugar. Okay, in RNA, each nucleotide sequence is an additional OH group at the second position of the ribose. And two nucleotides are linked through 3-5' phosphodiester linkages. Okay, phosphodiester linkages to form a dinucleotide sequence. When more nucleotides are linked, it forms a polynucleotide sequence. All these are very important. Look, instead of an H group, the RNA contains what? A hydroxyl group. And this makes a difference between RNA and DNA sugar structure. Okay, in this slide we have studied about the 3,5 phosphodiester linkage, dinucleotide, about the ribose sugar, and about the polynucleotide chain. Okay, phosphodiester linkages between the two nucleotide sequences. This is a very, very important thing. Okay, very, very important. DNA was an acidic substance. You know that DNA is negatively charged. You know that DNA is negatively charged. Yes, DNA is a negatively charged. Okay, DNA negatively charged, and it is an acidic substance. So the scientist was Friedrich Miescher in 1869. 1869, Friedrich Miescher just identified DNA to be an acidic substance. Negative, he was not the person. Okay. He just identified DNA to be an acidic substance. Okay, he just identified the DNA to be an acidic substance. And what does he did? He just identified DNA and named it as nuclein. Previous year NEET MCQ. NEET MCQ, okay? Friedrich Miescher was a person, very, very important. The very first scientist that you're studying in this chapter from NCERT. Friedrich Miescher in 1869 identified DNA and named it as nuclein. After that came James Watson and Francis Crick. They proposed the double helix structure of DNA. They proposed this particular structure, that is the double helix structure of DNA, was proposed by James Watson and Francis Crick. Okay, this is the person. James Watson, oh my God. Okay, this is a person. James Watson and Francis Crick. This is Friedrich Miescher. Friedrich Miescher. Okay, moreover, you can say that this double helix structure of DNA was proposed on the basis of an X-ray diffraction data produced by Maurice Wilkin and Rosalind Franklin. So, X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin. Very important again, NEET MCQ. NEET, AIMS, AIPMT, everything important question. Okay, this is a person, Maurice Wilkins and Rosalind Franklin. Okay. Next one. Structure of a DNA, or what are the salient features of the double helix structure of DNA? So the first point from that is DNA is made up of two polynucleotide chain. First point. You all know that DNA is composed of curved structures. They are twisted in a right-handed fashion, consists of two complementary sequences running anti-parallel polarity, running parallel and having an anti-parallel polarity. If one has 5 3 polarity, then the other will be having a 3 5 polarity. Look, if this is having a 5 3 the other will be having a 3 5 polarity. Since they are being complementary, they just combine together to have a nucleotide sequence or a double helix structure of DNA. Okay, DNA is made up of two polynucleotide chains coiled in a right-handed fashion. The pitch of the helix is 3.4 nanometer or 34 Armstrong. Previous year NEET question. Very important. Okay. For NEET 2020, this question will be there. Pitch of the helix will be 3.4 nanometer or 34 Armstrong. The number of base pairs in each turn is 10. Number of base pairs in each turn is 10. And the distance between adjacent base pairs is 0.34 nanometer or 3.4 nanometer. Very important. This particular slides contain 4 MCQ. 4 previous year MCQs from this particular slide. Okay, 4 previous year MCQ. 4 previous year MCQ will come from this particular session. Okay, very, very important. Okay, so let's move with the next one. Fifth feature, the backbone of the DNA is formed by sugar and phosphate. Very important. I have already told you. NEET, previous year MCQ. The backbone of the DNA is formed from sugar and phosphate, and what you can see is that the the DNA sequence is just project, not the DNA. The nitrogen base is just project to project from inside. The two chains have an anti-parallel polarity. One has a 5 3 polarity, and other has a 3 5 polarity. Okay, so look, how is it arranged? So this is two nucleotide sequences. This is two nucleotide sequences. 3 5 5 3 straight. They are twisted in a right-handed fashion. Twisted in a right-handed fashion. Okay, they are twisted in a right-handed fashion. Okay, next one. The structure of DNA. Okay, the structure of DNA. The two bases are paired through hydrogen bonding. Okay, the two bases in two strands are paired through hydrogen bonding, and you call it as base pairs. Okay, so the two bases, two strands, 1 second. I think my pen was not working, and now I just corrected it. Let me check it off. Yes, it's working. Yes, the bases in two strands are paired through hydrogen bonding. Okay, hydrogen bonding. A to T bond is 2 hydrogen bonding, and C to G bond is 3 hydrogen bonding. That is, adenine pairs with thymine using 2 hydrogen bonding, and cytosine pairs with guanine using 3 hydrogen bonding. And this pairing rule was put forth by Erwin Chargaff. Erwin Chargaff. You need the spelling? Yes. Take down. Erwin Chargaff. Okay. Erwin Chargaff was a person who just put forth this particular pairing theory. Okay, pairing theory. Not pairing theory, pairing theory. Okay. Purine comes opposite to pyrimidine. Purine comes opposite to pyrimidine, and this generates a uniform distance between the two strands. What was the pitch of the helix? 3 point. What is this, my dear children? 3.4 nanometer. Distance between two base pairs is 0.34 nanometer. Okay. Then let's move with the next one. Structure of DNA. This is a very important question from your NCERT. NCERT. One second. NCERT. NCERT textbook. Very important. Before going for your NEET, you have to look through this diagram. Before going to your NEET, you have to look through this diagram. Very, very important structures. You may ask about this base pairs. It will be easy for you to identify. They may ask some tricky questions from this particular slide. Okay. Nothing so important for me to just explain over here. One second. Erwin Chargaff rule. In DNA, the proportion of adenine is equal to thymine and guanine is equal to cytosine. Very important. The problem area of this chapter. That is not the problematic area. Okay, not the something which makes you very confusing. It is not like that. It is the part of the chapter or bio from which you may expect numerical questions for your NEET. So till 10th standard, you have studied numericals only in physics. When you come to plus 1, you see it in chemistry. When you come to plus 2, you see it in biology also. That is a very important word. Twist about NCERT and plus 1, plus 2. Okay? 11th and 12th. So adenine plus guanine must be equal to thymine plus cytosine. For example, you just have adenine divided by thymine is equal to guanine divided by cytosine is equal to what? This is simplest representation of Erwin Chargaff rule. Okay, or A plus G divided by T plus C is equal to 1. That is Erwin Chargaff rule. Okay. Very important. Okay, two things from this will be asked. Okay. Several times NEET questions have been from this. E. coli and haploid content of human DNA. E. coli, it has the DNA that is 4.6 into 10 raised to 6 base pairs in E. coli. You may ask numerical question from this. Haploid content of human DNA is 3.3 into 10 raised to 9 base pairs. E. coli has 4.6 into 10 raised to 6 base pairs, and haploid content of human DNA is 3.3 into 10 raised to 9 base pairs. For example, in phage bacteriophage, you have 5,386 nucleotide sequences, and the number of base pairs or the nucleotide sequences are the characteristic feature of an organism. Bacteriophage lambda has 48502. How much? 48,502 base pairs, which is a characteristic of that particular organism. Okay, so let's look what is there in the next one. Yes, this is a type of problem that they may be asking. Okay, this is a kind of problem that they may be asking. You may be asked to identify the length of a DNA sequence of human. You will be asked a question like this. Calculate the length of DNA of human. So you must know what the formula for calculating the length of DNA is: the number of base pairs into distance between two adjacent base pairs. This distance between two adjacent base pair, you know, 0.34 0.34 into 10 raised to minus 9 meter. Or from your text you have studied 0.34 nanometer. Okay, so the number of base pairs in human you have to by heart. How much is the number of base pairs? It is 6.6 into 10 raised to 9. And the number of distance, or the distance between adjacent base pairs is 0.34 into 10 raised to minus 9. So the length of DNA is multiply the product. 6.6 into 10 raised to minus 9, 10 raised to 9 into 0.34 into 10 raised to minus 9, you will get 2.2 meter. If you are asked to calculate the E. coli, then the length of DNA is 1.36 into 10 raised to minus 3 meter. And the number of base pairs is, okay, you are given the length. You are given the length as 1.36 and you are asked to find out, you know, the distance between two adjacent base pairs is 0.34 into 10 raised to minus 9, right? So you are asked to find out the number of base pairs. So you have to divide the length of DNA sequence by the distance between two adjacent sequence. So you'll be getting 4 into 10 raised to 6 base pairs which you have studied, right? I'll show you that. Okay, we have studied this length and this point in the last slide. Look, 4.6 into 10 raised to 6. That's approximately calculation. We get 4 into 10 raised to 6. So from this slide onwards, it's a packaging of DNA helix we'll be discussing in our next class. Okay, about the prokaryote packaging, then the eukaryote packaging we'll be studying. Okay, then we'll be studying about the nucleosomes, their arrangement in both organisms, okay, in prokaryotes and eukaryotes. Okay, thank you all. Please don't forget to like, share, and subscribe our wonderful channel. Till then, we meet with the next session of Molecular Basis of Inheritance. It's bye from your favorite channel, BioPoint. Okay, stream the world of bio.